Thank you. So I want to say first off that I sit in the sample tech group in the applications team, but I also work quite closely with people in the research and development groups. So a big shout out to them. They do some really amazing work. One of the questions that we get quite often is, are we going to get any updates or upgrades to the direct RNA kit anytime soon? And my answer to that is, well, how soon is now? Song reference. Anyway, so for those of you who don't know anything about direct RNA sequencing, or maybe you're not as familiar with it, it's unique. We are the only platform where you can actually sequence and base call RNA directly. We don't have to convert to DNA, which means we are not impinged upon any sort of biases that come from reverse transcription or PCR amplification. And we also maintain any sort of modifications excuse me, that are in RNA because we aren't converting to DNA. So it's unique, and I think it's pretty amazing. To give you a little bit of an understanding of where we are with the kit came out in 2017. We've had a couple of upgrades since then. At one point, we were working on chemistry called RNA3, which we were working on. It looked pretty promising, but we weren't quite comfortable with actually releasing it until we took a step back and reassessed and started working on a different chemistry, which is RNA4, which is what I'm here to talk about today. So RNA4 is a new kit that will be coming out. The sample prep is very similar to RNA2, so we start off with a 3' end ligation, which then we do a reverse transcription step. And the reverse transcription is only there to create a DNA-RNA hybrid molecule, and that's sort of to provide a scaffold and to sort of get rid of secondary structures to sort of help the molecule as the RNA is translocating through the pore. So you can see in the animation up there, that would be the RNA strand that's actually translocating, and that's what we're reading and base calling. The cDNA strand it never goes through the pore. We don't read it. It just peels off above the pore. So what we're base calling actually is the RNA. So the sample prep for this is very similar as far as inputs. We recommend somewhere between 100 and 200 nanograms of enriched RNA that can be poly-A selected or ribodepleted, or 1 microgram of total. You can go lower. People ask me this all the time. You can have lower inputs. You just have to understand that you're going to get less output. Now, one of the key features of this new sequencing chemistry is that we have a new RNA-compatible pore. So it's a pore that we're calling RP4. Which means it's going to have its own dedicated flow cell. So this is specific for, at the moment, RNA sequencing. And we also have a new faster motor, which is processing RNA at about 120 bases a second, which is an increase above what we are now with RNA2, which is the current kit, which is at 70 bases a second. We're now much faster, so we have increased output, which is very good. And then importantly, we've improved the accuracy by sort of incorporating a larger neural network, expanding the data set for training diversity. And so now we're reaching about 96% accuracy, single read accuracy, for something like a human transcriptome, which is a significant improvement, which is great. So as I said before, we have higher output because of the faster motor. So you can see that RNA4 in blue, RNA2 in gray, so we're reaching upwards of about 30 million reads from a PromethION flow cell, which is, we're getting into transcriptomic levels. This is great. So obviously, we also have other options for how you do the kit preps. You can do, as I mentioned, you can do poly-A, something that's enriched. That we generally get better output from something that's enriched as opposed to something like total RNA. However, if you can't get the amount of enriched sample to start a prep, you can actually do total RNA. It's fine. The prep will self-select for anything that's polyadenylated, and it works just fine. You can also do single-stranded. Some people don't want to or can't use the reverse transcription step. You can do single-stranded preps, but you will get a reduction in output, and that's something that we're continuing to look at. We test these, any sort of kit, we want to make sure that the data quality is good. So we test these with quantitative RNA panels, transcripts. And if we look at the known concentration of the representation of known concentration of molecules versus the observed, so we have expected versus observed, we get really high correlation, which is good, which means the data quality is nice. And these are in a range of lengths and representation. And then also, when we compare RNA2 to RNA4, we also continue to get a very nice correlation. So the new kit is also as good as RNA2 for that kind of data quality. And you can see that with long reads. We can see, again, RNA2 in gray, RNA4 in blue. You can see that for these particular examples, where we get nice long reads, so you get lots of gene coverage, and good isoform depth of coverage, so you can discern different isoforms from a particular gene. Of course, because it's RNA, we work in replicates, so we need to make sure that the kit is robust in producing that good quality data. So if you look at comparisons or correlations between two different replicates, you will see very high correlations. So the data quality is good in technical preps, typical replicate preps. And when we look at correlations between RNA2 data, this is human transcriptome data, we see the correlation is a little bit lower because we're looking at gene counts here, and the reason for that is because we actually have more genes present in the RNA4. It's better coverage, and so therefore it sort of pulls the correlations down in a good way. So this is good. And when I was saying about getting a better look or a deeper look at transcriptome representation, you can see that here in this violin plot in the middle. Again, RNA2, the current kit that we've got, which I now think of as old, is on the left, RNA4 is on the right, and if you look at gene count representation in those violin plots, you can see that the number of counts has increased for each. This is like a handful of genes that we use as sort of a metric. And I want to point out that there's a long non-coding RNA called exist that we see in very nice representation for the RNA2 prep because we have better output. And in RNA2, it just doesn't exist, right? So we didn't generate it from those data sets. So again, much deeper looks at transcriptomic data. Now, I'm running out of time. In fact, I'm out. So there are more things to come. I don't have time to talk about them, but I wanted to say we'll be talking about modified bases later in the Nanopore updates talk, so don't stick around for that. We are working on software improvements. Barcodes, we are working on them. We've had to sort of redesign them since RNA3, so it is something that we are actively working on. At the moment, the kit has been in beta testing stages, which Martin will be talking about because he's one of them. But we will be opening up for early access in, this QR code at the top is going live today, so you can register your interest for that. And then, lastly, what I wanted to say is that the kit's been out for 6 years, and people are doing things that they've never been able to do before. So people have never been able to look at RNA sequencing, specifically just the RNA. So there's been a whole new, and especially right now, RNA biology is just exploding, and there's a lot of interest in it. And there's been a lot of people that are doing some fantastic work that they've never been able to do before, particularly in things like modified bases or looking at things in infectious diseases, mRNA vaccines, and stuff like cancer research, which leads me on to Martin's talk. So Martin will be coming up here to talk about using RNA4, the new kit, and looking at that as far as functional profiling of cancer-associated long non-coding RNAs. But I also want to point out that there are other talks here. Mitten Jain will be in the presenting something in the Secret Cinema directly after this. And then we've got also Tsutomu Suzuki. And then later we have the RNA updates from Oxford Nanopore, which will be exciting. There'll be more direct RNA stuff in there, particularly for modified bases. Very exciting. And also all the other stuff that we have that's always amazing. So with that, I would like to say thank you and bring Martin to the podium. Thank you.
Thanks for entrusting us with this very preliminary data and technology. So I'll go straight into it. Our first runs that we did with the RNA004 flow cells, we tested just commercial RNA, poly-A enriched, and we got 14.75 million reads, which knocked my socks off. I thought, this is amazing. This is the update we were waiting for. And then we tested and compared it with total RNA as well to kind of get rid of that poly enrichment step, and we got very similar outputs, about 14 million reads. You can see here the pore occupation plots are really good, so it seems that the new pores are managing to stack up quite nicely. Relink distributions are pretty much what you get from your inputs. You might notice here that in our hands, the poly enrichment tends to drop the relinks a little tiny bit, probably because of the extra manipulation that needs to go into it using the polyase selection protocols. In terms of the quality, so once you map the reads to the human genome and look at the quality of the alignments and the base calling, it's amazing, 95, 96%. And you can also see that there's a slight increase in increase in accuracy when you use in vitro control or in vitro transcribed RNAs. So that error rate is probably due to the presence of RNA modifications. This is an example in a single exon, a long exon of what the reads look like and what their quality looks like. There's no filters here in IGV. This is every SNP, every indel is illustrated, and there's a 10% variant allo frequency on the consensus. So it looks really amazing. It looks like the R10 data for genomes. Of course, there's still these the small homopolymer issues, which you can easily get around, I think, with raw signal analysis. If you compare the size of the poly A from the raw signal, you can see that it correlates perfectly with what you would expect to get at 120, 125 bases per second. In terms of the raw data, it looks much cleaner, I find, than the RNA2. So the squiggles on the bottom, if you're used to looking at squiggles, you might notice there's a bit less noise in the squiggles. So we then asked, well, what can we do in a very short time frame with this new technology? What can we look at? And my longstanding interest is trying to understand non-coding regions of the human genome, particularly using RNA as a template for that. We know that about 2% codes for protein, and yet with high-throughput sequencing, it's now obvious that a lot more of our genome is biochemically active, predominantly through the production of non-coding RNAs. And the amount of non-coding RNAs that are indexed in reference databases has pretty much been constant over the past 10, 15 years. In fact, long-read sequencing has kept that going, and I think that the real amount is going to be quite higher. But long non-coding RNAs are challenging to experimentally and functionally validate. We know they work in a lot of different components of the cell. They do all sorts of regulatory roles and even functional catalytic roles. But when you find one that's in your condition or your disease, it's very hard to figure out what it's doing without throwing a whole battery of tests at it. So my colleagues at St. Justine, we are a pediatric hospital, so we do a lot of pediatric oncology. They've identified hundreds of long non-coding RNAs that are deregulated in leukemias, and they've compiled a list of all these genes and set out to do a CRISPR interference screen using a CRAB9 modified dead Cas9 protein, which essentially inhibits transcription and forms heterochromatin at the targeted loci. So they designed over 12,000 guide RNAs to target all of these long non-coding RNAs and perform the screen using a leukemia cell line and sequence the guide RNAs, the CRISPR plasmids over time to measure which long non-coding RNAs go up and down. On the right here, you can see some single molecule FISH data for 4 of the candidates that came out. So these are long non-coding RNAs that, when they were inhibited, reduced cell proliferation. You can see that the top 2 co-localized to P-bodies in the cytoplasm, and the bottom 2 co-localized with nuclear speckles in the nucleus, so potentially involved in splicing. So this is at the level of DNA, so you could be just causing heterochromatin formation and changes in gene expression this way. So they did a complementary follow-up experiment using CRISPR-Cas13 to go and perturb RNA directly by cutting it out, so essentially a knockdown assay. What they found was that indeed, choosing 5 of these candidates, 4 of them were knocked down quite easily, and 1 of them seemed to still be somewhat present. And it turns out that there's another isoform of this long non-coding RNA that was not targeted. And of course, when you do the knockdowns, you get the similar phenotype that was observed during the screen, suggesting that these are indeed important for cellular proliferation. So then we sequenced 20 flow cells of an RNA4, and these are the yields that we got. So you'll notice that we washed the flow cells for some of the runs and managed to get a lot more data out of them. And when you overlap them with the nature of the input, in our hands we had more data from the total RNA than with the poly-A RNA. And I think the reason for that is simply the library concentration. You get a lot more RNA with the total. You don't need to you don't lose any molecules in the cleanup, so your libraries are more concentrated. You can load less, you can reload if you need to. This is just showing that at the same number of pores, you get slightly higher output with the total RNA, or a similar level. And here is an example where you see all of the total RNA samples had a much higher concentration. So how do we analyze this data? We decided to use, Simona used the term isogenomics. I'm going to copyright it right now, isotranscriptomics. The idea is to generate a de novo transcriptome for your samples that you then quantify with your reads to make sure you're not introducing noise or missing data. So we did a de novo assembly of all the data that was generated using string tie2, and what we got was several thousand new isoforms. This is not surprising to us. We see this every time we do long-read transcriptomes. And when you use a synthetic control library and do the assemblies, you get similar kind of representation of the types of transcripts. And the fact that you get a lot more of the U classes, which are novel long-encoding RNAs or novel unknown loci, and some of the other more discrete categories of transcripts suggest that the references are essentially very incomplete. And yes, there's a bit of noise in this that still needs to be ironed out, but there's a lot more transcripts and a lot more diversity in splicing that we're missing with reference transcriptomes. Some examples. So this is a Alocus. If you look at the second last row in IGV here, you'll notice that the splicing patterns in this gene, I forget which gene it is, it's not important. What's important is look how clean the boundaries are between exons and introns. We did not see this with RNA2 or cDNA, the earlier cDNA runs. You can pick up all of the isoforms that are in your sample quite cleanly. On the bottom is the GenCode reference, the latest one, so you'll see that there's a lot more stuff in there that's not picked up. Another example here of a new long non-coding RNA that wasn't annotated in GenCode. We see quite a few of these. This one's quite highly expressed. And here's another example of a MALAT1, which is a very highly or cancer-associated RNA. It's very abundant in cells. And yet, you can quite clearly see here that there's a second isoform that's kind of in the middle of the transcript, of the reference transcript, that isn't well represented with the reference databases. So there's more to the data than meets the eye. We then use this reference to do differential gene expression. So we quantified the transcripts using Salmon. And you'll see here, when you look at the batch effects, there's indeed a difference between total RNA and poly-A RNA. And I believe the reason why is that we tend to get slightly smaller transcripts with poly-A enriched, which might affect the quantification of certain isoforms, particularly the 5' ends of them. If you apply batch correction, you can get rid of that quite easily and still be able to do differential gene expression. Here's an example of 2 of the long non-coding RNAs that we analyzed. The other 3 didn't give the drastic differentially expressed genes. Gene expression basically confirms the anti-proliferative phenotypes that we see. So there's stuff about cell proliferation and cell division for long non-coding RNA A, and for long non-coding RNA E, we see the top go hit is positive regulation of cell cycle. And then there's also some funny stuff like behavioral response to cocaine, which makes us wonder what they were doing in the lab when they were growing these cells. This slide, we found this yesterday. So we're looking at one of the top differentially expressed transcripts. And it turns out that it's, if I zoomed out a bit in IGV, and it's right next to KRAS, which is a notorious oncogene. This long non-coding RNA goes up when you knock down the long non-coding RNA E. So it's basically we're playing whack-a-mole with these long non-coding RNAs. You knock one out, and then a whole bunch of other stuff happens. Other long non-coding RNAs pop up. But what's interesting here is that it's a super well-studied gene, and yet there's not a single transcript that's annotated in this region in the reference genomes. So this is a really exciting finding, and we're going to probably chase this up in the upcoming weeks. So I'm out of time, but a big thanks to the lab. This was we brought everybody together and just threw everything we had at this over the past couple of weeks. We only got the flow cells, I think, 3 weeks ago, the first batch. So it's been a monumentous effort, especially by Bastien, Sean, Melanie, and Angel. Also, big thanks to collaborators at CHU Saint-Justine, Danielle Sinez lab, Claire and Alain and Manon did a lot of the CRISPR screens with the help of Richard Marcotte at the Canadian National Research Council. Eric Lecuyer's lab, Veneta in particular, did the single molecule imaging, and Martin Sauvageau gave the CRISPR-Cas with stuff. And a big thanks to Libby and Etienne, as well as our local Nanopore people for entrusting us with this data. And we're really excited about RNA004. I think it's going to change how we do transcriptomics and how we think about the genome. Thank you.